Today, I'm going to take you all in the ocean of hormone, especially histology of all the endocrine glands. That is very important to study for the point of view of your JRF, SRF examination, and also for veterinary surgeon exam also. This chapter is very important. So I will give you insights into the endocrine glands. First of all, we have to understand that what are the endocrine glands. Everyone knows who have done their BBSC and AH, they know well that endocrine glands are ductless. They secrete their secretory product directly into the bloodstream. So basically, the endocrine organs are hypothalamus, hypophysis cerebri, that is also known as pituitary gland, epiphysis cerebri, that is also known as pineal gland, because it is present on the dorsal surface of the cerebrum, in between the cerebrum and cerebellum. So this is pineal gland, thyroid gland, parathyroid gland, and adrenal glands. Guys, these are basic endocrine glands that secrete the hormones. And some other endocrine tissues and cells are also found in non-endocrine organs. See, for example, you have heard about the pancreatic islets. What are the pancreatic islets? Alpha, beta, delta, gamma cells. These islets are the part of the pancreatic exocrine part of the pancreas. And if I talk about the adipocytes, these are also the endocrine tissue and cells, but found in the non-endocrine organs. So adipocytes are present in the fat cells. And then cardiac myocytes. These are the muscles that are present in the heart. And juxtaglomerular cells of the kidney. These are also the endocrine cells that are present in the non-endocrine organs. Cells of the theca interna. If I talk about the cells of the membrana granulosa, interstitial cells of ovary, corpus luteal cells of the ovary, these all are the endocrine cells but are present in the non-endocrine organs. Some more examples are sustentacular cells of the testes and interstitial cells of the testes. So I'm going to discuss about all the endocrine and non-endocrine glands of the animal body. So before heading towards the endocrine glands, just take a look on their derivatives. So if I talk about the pituitary gland, pineal gland, these are derived from the ectoderm. And if I talk about the thyroid gland and parathyroid gland, these are endodermal derivatives. So now let's start with the hypothalamus. Everyone knows that hypothalamus is a big name in the if I talk about the endocrine gland. So hypothalamus basically contains some neurons and some nucleus that are paired. See, for example, supraoptic nuclei, you can see here, this is the supraoptic nucleus, and paraventricular nuclei and arcuate nuclei. And the most important nucleus is suprachiasmatic nucleus. Suprachiasmatic nucleus. It is very important if I talk about the pineal gland. And later I will discuss about this suprachiasmatic nucleus. And this hypothalamus is very much required to blood volume and the osmolarity. And next, start with the hypophysis cerebri. Hypophysis cerebri, everyone knows who have done their BBSC and AH, that it has two parts, adenohypophysis and neurohypophysis. You can see this figure. This is the part of the adenohypophysis. This is the, because adenohypophysis has three parts, pars tuberalis, this is the pars tuberalis, and this is the pars distalis, and pars intermedia in between these pars distalis and pars nervosa. So it has three parts. So let's talk about the which part is the largest part. You can see that the largest part is the pars tuberalis. And guys, this part has a vast diverse cell population. I will tell you in my later slides that what are the cells that are present in the pars distalis and other parts of these of this adenohypophysis and neurohypophysis. And this is the pars intermedia. This is present at the junction of the adenohypophysis and neurohypophysis. There and neurohypophysis. This is the neurohypophysis. And neurohypophysis, you can see that it has two parts, infundibular stalk and pars nervosa part. And this, the part that is present on the adenohypophysis, it is connecting the median eminence. You can see this, this is the median eminence. If I talk about the pars tuberalis, as in the figure, you have seen that it connects the adenohypophysis with the median eminence. And it forms a collar. It forms a collar around infundibular stalk of the neurohypophysis. And if I talk about the neurohypophysis, if I talk about the its embryology, how it develops as a midline imagination of the floor of the hypothalamus, and it forms a funnel-like infundibulum proximally. Funnel-like infundibulum ये आगे की तरफ proximally and pars nervosa distally. Distal है तो इसको distal बोल दिया. Now, because our topic will be focused on the histology of endocrine glands, so this is the histology of the pituitary gland. You can see the hypothalamus, stalk of the pars nervosa, lumen of the Rathke's pouch. Pars nervosa part is being shown to all of you, and this is the pars distalis. And this gland is wholly covered by the capsule, and it chiefly contains collagen fibers. And you may find elastic and reticular fibers sometimes, but most of the time you will see collagen fibers. And for the collagen fibers, what stain we will perform? We will perform Mason's trichome stain. If I talk about the pars distalis parenchyma, it contains cluster of cells that is channelized by the sinusoidal capillaries. You can see the sinusoidal capillaries, and these cells that are present in the pars distalis are present in the form of the cluster. You can see this is the cluster, this is the cluster. And basically what types of cells are present? Chromophil cells and chromophobe cells. And these chromophobe cells form more than 50% of the cell population in the pars distalis. And chromophil cells, if I say about the chromophil cells, there are two types of cells, acidophil and basophil cells. In some species, if I talk about the buffalo, then this pars distalis is divided in central zone and peripheral. And this question was asked that in buffalo, the pars distalis is in which species pars distalis is divided in case of. So the answer is buffalo, the central zone and peripheral zone. And this central zone is highly vascular. Vascularity, how we will see the vascularity? There will be more and more blood vessels, arteries and veins will be present. And it contains higher concentration of basophils and chromophobe cells. And if I talk about the peripheral zone, it is less vascular and it is rich in acidophil cells. You can see here, acidophil okay, somatotrophin, growth hormone, and most of on this slide you will see the chromophobe cells, and basophil cells are also present. And apart from these cells, if we go to immunohistochemically, so we will find the 5 types of subtypes of cells. First one is the somatotroph cells, lectotroph, thyrotroph, gonadotroph, corticotroph, right? And these are the, and acidophil cells are other part of the cell that includes somatotroph and lectotroph, and basophil cells include thyrotroph and gonadotrophs and corticotrophs. Now let's look into cells form the more than 50% of population of cells in the in this gland, and it vary in appearance and location and shape. And some cells are simple epithelium, and some cells having cysts of unknown significance, and some cells are stellate shape also. And many different authors told about the shape of these cells in a different way. So stellate shape, you will learn stellate shape cells are present basically. And if I talk about the pars intermedia, so in case of horse, this pars intermedia is well developed, and it may form, and it may be form of a simple columnar epithelium or pseudostratified columnar epithelium. You can see here how the pars intermedia is present here, and this is the mesotrichome stain that is being used in this diagram, and you can see the collagen fibers here, and this is the pars nervosa part. You will also see in the histology of pituitary gland many sinusoidal capillaries, and chief cells will that and the chief cells that will be present in these are melanotrophs, and they will secrete melanocyte stimulating hormone MSH and B lipotropin hormone. Everyone must have known about the melanocyte stimulating hormone, but I think B lipotropin hormone you may not have heard about this. So this hormone will also be secreted by the pars intermedia, by the melanotroph cells. Now let's talk about the pars tuberalis. You have seen this the diagram that pars tuberalis, how pars tuberalis was present as in the form of a collar around the infundibular stalk. So let's just imagine this that figure. And these, if I talk about the pars tuberalis, it chiefly consists of low columnar cells. And these low columnar cells will be present in the form of a cluster that uh, uh, I have made you seen the cluster in my previous slide. And these cells are melatonin receptors. And these receptors participate in regulation of seasonal reproductive cycle of some domestic mammals. You will learn this. And if I talk about the pars nervosa, I have told you that it consists of median eminence, infundibular stalk, or stem, you can say, infundibular process, or pars nervosa. And it is also comprised of nerve fibers, pituicytes, and blood vessels. And if I talk about the infundibular cavity, it is the extension of the third ventricle, and it is covered by a wall of the infundibular stalk. This is the infundibular stalk, and you will learn this question will be asked in somewhere or in future in other exams, that this infundibular stalk will be uh, covered by the single layer of ependymal cells. And then this question will be many times will be asked that ependymal cells are present in. Okay? This is the histology of pituitary gland. Now, let's talk about the pineal gland. The pineal gland, that is known as pile capsule, and pineal gland consists of pinealocytes and astrocytes. The main glial cell that will be present in the pineal gland will be astrocyte, but pinealocytes form the 90%. But in case of human beings, 90% population in case of human being. But in case of animals also, pinealocytes will be present in a higher amount. And these pinealocytes, how will we differentiate pinealocytes from the astrocytes? You can see these pinealocytes will be larger than the astrocytes, and pinealocytes will be lighter in color as compared to the astrocytes. So this is the difference. And the pianocytes will be larger, and these rounded nuclei is surrounded by a broad rim of light cytoplasm. This is the boundary of the cell. It is very faint, And this is the nucleus of the pinealocytes, that is rounded nuclei. And astrocytes will be dark in color, small in size, and having darker nuclei. This is the darker nuclei as compared to the pinealocytes. So these both pinealocytes and astrocytes have long processes which form neuropil between the cells. These processes, you will see these processes, but with the help of other stains. See, for example, Ramens and Kegel's method. By this method, you will see the processes of pinealocytes, but not with the help of H&E stain. And let's delve into the parenchyma of the pineal gland, and you will see the numerous capillaries that will be transmitting the pinealocyte secretions. And you will also see some pericapillar phagocytes, which act as antigen presenting cells. And if I talk about the brain sand, that is a part of the pineal gland, you will see only and only in the histology of, yeah, this question will be asked in many exams. Brain sand is present in the pineal gland, but in the mammalian pineal gland. Not in case of birds. Brain sand is totally absent in case of birds, reptiles, fishes, but it is present in case of mammals, right? And these are the calcified. Now the question will be asked that what are the corpora arenacea? What how it is made up of? It is made up of calcified protein deposits. And in addition to these structures, its parenchyma also have neurons in higher vertebrates and neuronal-like peptidergic. You know very well from your first year of your BVSC, you will be listening this that pineal gland is directly or indirectly involved in the daily or seasonal photoperiodically induced rhythm in sexual reproduction, in sexual behavior, in thermoregulation, and in color changes. And it also regulates secretory activity of the pinealocytes. If I talk about in some seasonal polyestrous domesticated animals, the pineal activity resulting in that I have right now here. Pineal gland has many dark blackish cells having melanin pigment along of pinealocytes in horse. If I talk about the histology of the horse pineal gland, you will see large number of ergyrophilic substances inside the pinealocytes. And pinealocytes will be present in the pineal gland. But in case of older animals, some species, in case of yellowish lipofuscin pigment, that will also be present in case of in the histology of pineal gland. But in case of pigs, uniformly distributed, you will see uniformly distributed mucopolysaccharides in the cytoplasm of pinealocytes and glial cells. But in case of pigs, if I talk about the parenchymal cells of the rat pinealocytes, then it will contain large amount of oxidative enzymes and non-specific esterases. Less leucine amino peptidase and acid phosphatase will be present in the parenchymal cells of the rat pinealocytes, and high concentration of the alkaline phosphatase will be present in the wall of the capillaries. These are the some research points that someone has done their research pinealocyte that I have mentioned here. And now let's talk about the thyroid gland. Everyone knows about the thyroid gland, that how it is important in a human being also and in case of animals also. How these thyroid gland, if we go into the histology, it is surrounded by a dense irregular connective tissue capsule. This is the capsule that I'm scrolling my arrow, you can see the capsule. This is the capsule of the thyroid gland, and in this capsule, and there is a septa that is dividing the thyroid gland. You can see that these are the septa that are running from the capsule and is dividing the thyroid gland into many interconnected lobules. These are the lobules, these are the septa, these are the lobules, and lobules consist of vesicles of varying sizes thyroid follicles. You can see the varying sizes thyroid follicles. Some are large, some are medium, some are very small. And fat-free interfollicular connective tissue have a network of delicate reticular fibers. Reticular fibers you will not see in this diagram because some other stain will be performed to show these reticular fibers. Some silver stain will be performed for the reticular fibers, but in this diagram you will not see this. This is only for the information, and you can see in the blood capillaries are present in the capsule. And if I talk about the follicle, if you see the inside the follicle, this is the follicle, and inside the follicle you will see the colloid. Colloid is made up of protein. Colloid is made up of protein. And in between these follicular cells, you will see the vacant space in which or parafollicular cells will be present. These are the nucleus of the parafollicular cells. You can see my mouse arrow. This is the in between the follicular cells, parafollicular cells will be present. And if I talk about the epithelium of the follicular cells, it will be simple squamous or simple cuboidal or simple columnar. It depends. In case of in resting state, or in case of inactive state, the follicular cells that are surrounding the, but in case of its activity in active state, it is made up of simple cuboidal cells. Parafollicular cells, if I talk about, in case of dog, it is very prominent. You can see these are the parafollicular cells, and it is positioned between the, I have told you earlier in my previous slide, the basal lamina. Also from it, they also form cluster of 5 to 6 cells or 4 to 5 cells, and they they are large amine precursor update decarboxylation cells, and they synthesize calcitonin. This question will be was asked to me in my interview of assistant professor, that parafollicular cells secrete they synthesize the calcitonin. And now parathyroid gland just present beside the, just below the thyroid gland. If we cut the section of thyroid gland, and we see the histology of thyroid, then automatically we will see the histology of the parathyroid gland also, because this tissue will be adhere to the thyroid gland. So that's why this parathyroid gland comes. And this is a thin capsule. You can see this is the parathyroid gland, but this is the thyroid gland. This parathyroid gland, you can see, and it is surrounded by a thin capsule, and it is made up of interstitial reticular connective tissue. If I talk about interstitial reticular connective tissue of the parathyroid gland, it is very heavy in case of dog and ox, and it is very delicate in case of sheep, goats, and young animals, and it contains abundant adipose tissue in older individuals, and some larger lymphatic pressure generation. Pacinian can see many cells here that are forming the cluster. And you can see the oxyphil cells and chief cells. And if I talk about the, uh, you can see that there are different cells, and this question will be asked that oxyphil cells are present in only and only in parathyroid gland, and parathyroid gland are in two pairs. Two pairs parathyroid gland are present in case of animals. And if I talk about the uh, function of the parathyroid gland, so everyone knows that it's secretion of it secretes a hormone, that is parathormone, and it regulates a calcium and phosphorus metabolism. And if I remove this gland, then what will happen? If we remove the parathyroid gland from the animals, it results in the lowered blood calcium, and fetal parathyroid tetany will be formed. Now let's talk about the part. It is very important to know this, that cortex part is derived from the coelomic mesoderm, and medulla part is derived from the ectodermal tissue or neuroendocrine tissue. And these two portions, if I अगर हम embryologically अगर हम बात करें, these two one organ, that is adrenal gland. And connective tissue, if I talk about its connective tissue capsule, it contains elastic and smooth muscle fibers. Most of the time, you will see elastic fibers in the adrenal gland capsule. And in case of pituitary gland, we heard that in the capsule, mostly collagen fibers was present, but adrenal gland capsule, elastic fibers dominate. So you can see this is the histology of the adrenal gland. You can see all the parts. Cortex part is being shown to all of you. And you can see, you have heard also in your BBSC course that the cortex has three parts. The first part, that is zona glomerulosa, it is present outer, zona fasciculata in the mid part, and zona reticularis in the innermost part. So we can say that from outside inward, the layers are zona glomerulosa, zona fasciculata, and zona reticularis. It is very important to know that between outer and inner cortices, a narrow band zona intermedia is also present. And it is small, having undifferentiated stem cells, and cortical cells of this zona intermedia region contain nuclei that are poor in chromatin substances. Now let's go into the zona glomerulosa, then what is the importance of this and how the cells are arranged in the zona glomerulosa. These cells are arranged in the form of a tuft of epithelial cells, and these are mostly columnar cells. And the source of this zona glomerulosa is mineralocorticoids. And unlike rounded mitochondria in the inner cortex, the mitochondria are elongated with lamellar cristae. These mitochondria you will see in the transmission electron microscope, not in the light microscopy. And cells from the zona intermedia differentiate and undergo apoptosis in this zone. If I talk about the organelles and inclusions of outer cortical cells, are very important and are as essential for the steroidogenesis. If I talk about the species like horsehead, and their polyglobulonal cells change to long columnar cells laid down transversely. Now let's talk about the zona fasciculata. This is the diagram that is being shown to all of you. It is a zona fasciculata. You can see the spongioscytes and blood capillaries are also present in this sinusoidal space. And you will see these cells in the form of a cord. These cells are arranged in the cords. You can see here This is the nucleus of the cells, and they produce glucocorticoids. And glucocorticoids, you know very well, cortisol and cortisone. This and form a network. And it is made up of a polyhedral cells arranged as a network of anastomosing cords and plates. And these are separated by a large sinusoids. Here you will also see sinusoidal means vacant space that will be separating some different lobules, zones, and zones will or lobules will contain different cells. That you can see these cells here. This is the nucleus of the cell. This is the cell, but the boundary of the cells is not very clear. But sinusoidal space are very clear. And these cells synthesize a small amount of androgens, and cells are oldest of inner cortex. Apoptotic cells are frequently observed in this. And cells of inner cortex migrate from zona intermedia and form the zona fasciculata and then zona reticularis. Now let's talk about the medulla part of the adrenal gland. It is mainly derived from the ectoderm that I have told you, and it is especially abundant in ruminants, and it synthesizes epinephrine and norepinephrine, and it stores these neurotransmitters as secretory granules, and fine net-like connective tissue framework contain elastic fibers in this. If I talk about the chromaffin medullary cells, it is very important to know that chromaffin cells are present in the histology of medulla region of adrenal gland. And what are the characteristic of these chromaffin cells? These form the anastomosing strands or clusters separated by a sinusoids, and they are आसानी से they are readily stained with the basic dyes and columnar, polygonal or columnar in swine and in case of sheep. You can see these are the chromaffin cells in the medulla region of the adrenal gland is present, and these cells show a fine chromaffin granulation. You can see inside this nucleus fine dotted structure is are present. It is fine chromaffin granulation is present inside the nucleus. You can see, and it varies with the adrenaline content of the gland. And in this nucleus is a large pale nuclei are located in the central portion of the cell cords. Now let's talk about the endocrine cells of the heart, cardiac myocytes. That these are the endocrine cells, but are present in the non-endocrine organ. Between 1971 and 1983, when polypeptides atrial natriuretic factor and brain natriuretic peptide was investigated, that was produced by the mammalian heart atria. But now this is known as the hormone that is secreted by the endocrine cells of the heart. It is known as atrinatriuretic uh, peptide, and it is associated with the maintenance of the extracellular. Talk about the other polypeptidal hormone that are expressed in the heart. These are C-type natriuretic peptide, adrenomedullin, proadrenomedullin, N-terminal peptide, and endothelin 1. This endothelin 1 was asked in some exams in some other year, that endothelin 1 is secreted by heart. And in kidney, what is the role of atrinatriuretic peptide? It acts in the collecting tubule to antagonize the effect of antidiuretic hormone, and it also acts on the juxtaglomerular cells to inhibit release of renin, and in case of brain, this ANP inhibits ADH release. Now let's talk about the juxtaglomerular cells of the kidney. These are also the endocrine cells, but are present in the non-endocrine organ, that is kidney. These juxtaglomerular cells are modified rounded smooth myocytes of tunica media of afferent arteriole, and it is basically present at vascular pole of the renal corpuscles. And these synthesize, store and release renin granules. रेनिन डबल एन है, हम बच्चों को बताते हैं कि डबल एन है तो एंजाइम है, सिंगल एन है तो हार्मोन है, RENIN, दिस इज द हार्मोन, and it is very important element of the renin angiotensin aldosterone system. We have learned many times in our BVSC course in case of in veterinary physiology department that what is the importance of renin angiotensin aldosterone system, and it is released in response to a baroreceptor signal, and baroreceptor signal is initiated by the lowered blood calcium and then sympathetic nerve endings found near juxtaglomerular cells. Now let's talk about the endocrine cells of the ovary. These are follicular epithelial cells. You can also say these granulosal cells, theca interna cells, luteal cells of the corpus luteum, and cells of the ovarian stroma. These all are the endocrine cells of the non-endocrine part, that is ovary. Now the follicular epithelial cells we will talk about. Basically, follicular epithelial cells are polyhedral, but its basal layer may be columnar. Its basal layer may be columnar. It is very important. And if we stay see the past positive inclusions in this, and these past positive inclusions is known as Cole-Exner bodies. So this question many times will be asked in the SRF also. Cole-Exner bodies are present in the ovary in, and if in, and ovary may be kahan pe, follicular epithelial cell, and they express the FSH receptor. These receptor, and FSH results in follicular cells proliferation and differentiation, and in large follicles induces expression of LH receptors on same follicular epitheliocytes. And if I talk about the theca interna cells, these are spindle-shaped cells that will be located in a delicate reticular fiber network, and they will express the LH receptors also. You can see the some blood vessels are present in this, and some thecal cells of atretic tertiary follicle remain viable as steroidogenic interstitial endocrine cells that are known as stromal organs. Now, corpus luteum, you can see the theca lutein cells here, and granulosa lutein cells, vessels, and large lutein cells, small lutein cells, you can see here. So corpus luteum consists of large luteal and small luteal cells. These large luteal cells have polygonal shape with large spherical vesicular nucleus, and they will contain the numerous lipid inclusions. These are the lipid inclusions are present. And if I talk about the small luteal cells, they have relatively more lipid droplets, and they will occupy mainly trabecular and peripheral area of the corpus luteum. Now, ovarian stromal cells. You will ask me that how ovarian stromal cells are different from the other cells. So these ovarian stromal cells differ from typical connective tissue as mostly it has spindle-shaped stromal cells. It has spindle-shaped cells, and abundantly it is supplied with the blood vessels and capillaries, and it is similar with the fibroblast cells also. And if I talk about the stroma of ovarian cells, it is containing connective tissue such as reticulum and collagen. And stromal cells are associated with the maturing follicles that may acquire endocrine function and secrete later estrogens. This is the ovarian stroma. Primordial follicle is being shown, primary follicle is being shown, and this is wholly a stromal ovarian stroma. You can see here. Now let's talk about the endocrine cells of the placenta. You all have heard in your gynecology that placenta also secrete some hormones, some synthesize some hormones, say for example estrogen, progesterone, ECG, PMSG, prolactin, and pro-opio-melanocortin derivatives also. But in case of mare, some trophoblast cells of the placental membranes migrate into endometrium, where they will be proliferating themselves, and they will be forming large clusters that is known as endometrial tufts that will synthesize the PMSG, means pregnant mare's gonadotropin. Now, let's talk about the endocrine cells of the testis. Two cells are present in the testis. The first one is interstitial cells, also known as Leydig cells, and sustentacular cells. You will see the interstitial cell just outside of this, right? Outside of this, and the susta within the seminiferous tubule. Sustentacular cells, also known as Sertoli cells, you will see inside the seminiferous tubule. This is the seminiferous tubule, and Leydig cells are present outside the seminiferous tubule. Or you can say that between the seminiferous tubule and within the seminiferous tubule are present Sertoli cells or sustentacular cells. That will release the spermatocyte, then spermatozoa, and these are the spermatogonia. You can see here in the periphery of the this this seminiferous tubule, and interstitial cells. You can see here interstitial cells outside the seminiferous tubule. So basically, leading cells are steroidogenic, and stable cells adjacent to the seminiferous tubules are present, and these are large polymorphous cells having spherical nucleus. You can see this is the nucleus that is having a spherical shape, and these occur in cluster. There are many cells are present that are present in cluster or cords in close contact with the capillaries. And if I talk about the sustentacular cells, the FSH receptor on sustentacular cells bind with the FSH, which results in synthesis of estrogen from endogens released by interstitial cells, and release peptide inhibin and androgen binding protein. These cells release peptide inhibin and androgen binding protein. This is the function of these cells. And testicular hormone targets secondary sex tissues like prostate, skeletal muscles, and they will be exerting a negative feedback on GnRH hormone that is released from the hypothalamic neurons, and FSH and LH released from the hypophyseal gonadotrophs. And if I talk about the inhibit, it will target the gonadotrophs, selectively inhibiting the FSH release. So these are all about the endocrine gland and their hormone histology functions I told you. So if there is any doubt, you can ask me any question regarding the histology of the endocrine glands. Now the lecture is complete. Any question?